I will now cover the last part of CiPA, which is the clinical ECG biomarkers part, which is definitely the most exciting part. And as well, in addition to that, the post-market risk assessment for Torsades. So the phase 1 ECG biomarker working group is led by FDA as well as the Cardiac Safety Research Consortium. And its goal is to use human phase 1 ECG data to determine if there are any unexpected ion channel effects compared to the preclinical ion channel data. So for a new ECG biomarker to be helpful, it would need to have additional information beyond just the PR interval, the QRS interval, or the QT interval. So that new ECG biomarker would need to differentiate between multi-ion channel effects during repolarization. It needs to have sufficient power to detect changes in small sample sizes with exposure response analysis, and it will need to be available for widespread use. So this slide is an overview of the 12 ECG biomarkers that have been tested so far. But the main point to remember here is that there was an exposure-response relationship present for many of these ECG biomarkers, and most of these paralleled the changes in QT without adding any additional information. So none of these biomarkers were actually good as a new ECG biomarker except for one, which I'll discuss now. So what you'll see on this slide is on the top a surface EKG with the QRS complex, the J point, and the T wave with a cardiac ventricular action potential below that. So what we can see is that the QRS complex is mainly made up of the rapid upstroke of the cardiac action potential with an influx of sodium. Then the J to T peak interval is mainly made up of the plateau phase of the action potential and late sodium and calcium influx, while the T peak to T end part is made up of the repolarization phase of the cardiac action potential with hERG potassium outward current. So when I then look at these two graphs, on the left, the selective hERG blocker dofetilide with dofetilide concentration on the x-axis and interval prolongation on the y-axis, we see that there is Q-to-C prolongation with a selective hERG blocker. And that is mainly made up of J to T peak prolongation as well as T peak to T end prolongation. Now, when we compare that to the right of a multi-channel blocker, ranolazine with ranolazine concentration again on the x-axis, that blocks late sodium as well as hERG potassium current, we see that there's Q-to-C prolongation. However, that's now mainly made up by T peak to T end prolongation, while there's no change in J to T peak in the J to T peak interval. So in summary, for the phase 1 ECG biomarker part of CiPA, we saw that J to T peak was identified as the strongest predictor of inward current block in the presence of hERG block. And what we've done so far is that FDA developed a J to T peak as well as T peak to T end algorithm that is being released as open source software. And there is a new prospective clinical phase 1 ECG validation study planned to validate the J to T peak biomarker, and that will hopefully be completed in 2017. And then I want to move on to the post-market risk assessment for Torsades. And as said by my colleagues, there were 28 drugs in CiPA, 12 used for training and calibration, and 16 for validation. And those were picked because of well-defined electrophysiology and known clinical characteristics, and were identified by a sub-team of experts. So these 28 drugs, as heard before, were then divided into high, intermediate, and low risk for Torsades based on published reports, analysis of the FDA Adverse Event Reporting System, or FAERS, other data sources, and expert opinion. So to potentially validate the CiPA paradigm in the post-market space, we need to consider that Torsades is a very rare event, and it's therefore difficult to detect. So to determine if there's a real-world Torsades risk for a certain drug, large databases are required. So FDA and other entities, such as the Credible Meds website, currently use the FAERS database to quantify this risk. So briefly, what is FAERS? FAERS is a database at FDA at which patients, consumers, and healthcare professionals, other patients, and other people can voluntarily report an adverse event that they think might be related to a certain drug. And that could either go through the FDA MedWatch program, which leads to 5% of all the reports in FAERS, or to the manufacturer, who is usually required to report it to FDA. And that pathway consists of 95% of all reports in FAERS. So even though this risk assessment using FAERS is a very sensitive approach, there are potential problems since a lot of the confounding factors that might influence the relationship between drug exposure and the Torsades event is not captured in FAERS. There's a causality issue. And we don't actually know for a certain drug how big the problem is because we don't always know how many people are using this drug. So there's a lack of denominator data in FAERS. So to mitigate some of these problems, we are currently using large databases of drug utilization, prescription, and comorbidity data that can provide a more accurate risk of Torsades by adjusting the FAERS report just mentioned for confounding factors and putting them into a drug utilization context. So that concludes the CiPA part. I just want to mention the expected outcomes of CiPA overall. CiPA is a standardized non-clinical mechanistic-based study to determine proarrhythmic risk that can be applied early in drug development to aid in compound selection. Proarrhythmic risk is calibrated against consensus clinical comparators ranked according to clinical experience. Compounds with hook block and QT prolongation that might be dropped from development currently could have a clearer path to advance if they are shown to not be proarrhythmic. QT prolonging drugs on the market currently that are not proarrhythmic could have potential labeling updates to reflect this. And the CiPA paradigm could be a potential model for comprehensive in vitro mechanistic-based approaches to be applied in other drug safety areas as well.